worked the first time, so I'm not sure.
Okay. Can you not get it off the presenter mode?
No, I don't know why it worked the first time, so I'm not sure why it's not like letting me switch it. So, okay, I think I got it now.
Okay.
Sorry about that. How about now?
There we go. All right, perfect. Let's go. All right, like just perfect time-wise. All right. Okay, it's 3:10. Yes. Welcome to the fall Iowa Nutrient Research Center seminar series. Thank you all for attending either virtually or in person. And I'm just going to let everybody in that's coming into in there. Okay. Matt Helmers with Iowa Nutrient Research Center. Again, welcome you all to the fall seminar series. In the past, I guess it would be 3 years ago, or over 3 years ago, when we started this in 2020, it was like the history of some water quality research throughout the Midwest. So we had a lot of folks that were more senior, I won't say old, but more senior. And then last year we had folks that PIs that had INRC funded work to kind of talk about what some of the impacts are. And, you know, really excited for this fall to hear from the next generation, the people that are going to make things happen in the future. It's not going to be some of us that have less time left in our career than we've been in our career. You know, I was thinking myself, I've been at Iowa State 20 years now. I don't think I'll be at Iowa State 20 years from now. I think I'll be retired and hopefully spending my time on a golf course or something like that somewhere in the US. So this fall, we're able to feature some former graduate students that worked on INRC funded projects, as well as some current graduate students. So we're breaking it up like we did last year in 2 presentations. So the first one, we're going to have Dr. Morgan Davis, that's now at the School of Natural Resources at the University of Missouri. Dr. Davis worked in Dr. Eisenhardt's lab while he was here for his PhD, and in Dr. Marshall McDaniel's lab for his postdoc. That might have given away what Marshall was going to talk about, or Morgan was going to talk about. So I'll turn it over to you, Morgan. And for those online, if you have questions, feel free to type those in the chat box, or we'll hopefully have time to unmute if you have a question, and you can relay it directly to Morgan. So with that, the virtual floor is all yours, Morgan.
Great. Thanks, Matt. And thanks everybody online and in person. This was a fun little presentation to put together because a lot of the work that I did with the INRC, I think, really helped me get my position where I'm at, and is still a big source of a lot of my collaborations. So also thanks to Malcolm for organizing all this and giving us kind of a structure to follow. So today I'm going to go over just a little bit about myself and then my current position, then go into some of the details of the work that a group of us did, essentially 16 through 18 to maybe even 2020, and then maybe touch on how INRC kind of helped me secure my current position. So my name is Morgan, as was said, and I did my bachelor's and master's at the University of Illinois with Mark David. So even though I was a soils focused project there, part of my research journey really started with water quality and looking at nitrogen specifically. And as Matt said, PhD and postdoc at Iowa State. I also have two little ones that are a lot of fun. And so we are going to miss summertime because it was we had a lot of fun at the pool, etc., with my two goofballs here. So so that's outside of work, that's pretty much consumes all of my time. So yeah, so in my current position, I'm an assistant professor in soil biogeochemistry and ecosystem sustainability is the very long, at least what I was hired under. So I here at the University of Missouri, like a lot of other institutions, Iowa State being kind of an exception for that one, the soils faculty split from the crops faculty, to kind of absolve an agronomy department, and soils went to natural resources with forestry and wildlife, and crop science went to form kind of their own department of plant science. We still do a lot of work with those groups. We also in Columbia house a USDA ARS station, and so those collaborations have been vital. And Dr. Lori Avendrock, who I also know was she also did her PhD at Iowa State the same time as me, is currently a research agronomist at the USDA down here, and so we're able to continue some of those collaborations. But in general, my position is 45% teaching, 45% research, and then 200% service is what bad joke, but that's what I like to joke about. So I'm essentially half teaching and half research. I get to teach a larger introduction to environmental science class that is for our majors in our environmental science undergrad curriculum, as well as a general elective as a general science elective. So it's a fun class to teach too. We have everything from ceramics and journalism majors to really smart undergrads that want to go to law school and be environmental lawyers. So it's a fun class. I also teach a pollutant, fate, and transport class. I co-teach that with another. I do the fate portion, and then I have a colleague that does the transport portion. And then I teach a soil biogeochemistry class for graduate students in the spring. So my research is mainly focused on, as much as I don't want to say, as I've pushed back from the term soil health, just because it seems like a grab for everything. That is a big focus of our component, a big component of our research focus here. But one theme that I kind of measured through a lot of our different systems is soil gas flux, and specifically following nitrogen from inorganic form in the soil of nitrate ammonium to gaseous losses, specifically nitrous oxide. So anytime I do a talk where I have get to talk to some Iowans again, I always like to pull up this ecoregions map of Iowa and Missouri, which is really fun for me to look at now with the research I've kind of done from north now to down in the Ozarks. So I've been lucky enough. I'm just going to highlight a couple things with the research program right now to continue to work with many of you that are in the room, even though I can't see you, with a USDA CAP project with perennial ground cover, the Region PGC project. So that's been a very fun project to work on. And so I've continued some work there with Marshall McDaniel, focusing more on the greenhouse gas side of the soils work. As we've kind of moved further south, we also have a few projects in northern Missouri looking at nitrification inhibitors and the potential effect of nitrous oxide emissions there. So we work closely with Kelly Nelson, who has been a collaborator with many of you I know up in Iowa State for a long time. So we're lucky there. We haven't completely gotten out of the water world. We're still doing some stream bank erosion work, and we do this actually close to Columbia. That is a pretty neat ecotone, a change from glaciated landscape to not. So that's a really fun area where kind of nutrients kind of collide in a lot of ways. And with these landscape position changes, there's some real promise for conservation practices, including riparian areas, as well as just changing overall what is planted in that area to begin with, whether you're going from a kind of row crop to potentially other commodity crops or even just forested areas. And then finally, the other really fun thing about being down in Missouri is getting to do some work with the Forest Service and in forest ecosystems in general. So this is one of my grad students, Will Rump, presenting here on looking at some conservation practices on log landings where they stack logs for timber harvests. So we're trying some different treatments, including biochar and ripping, to reduce the compaction that happens from those as well. So that was just kind of a brief highlight of what we have going on down here in Missouri in our program. Now I'd like to highlight the work that I did for my PhD, and none of this work would really be possible without Michelle Sapir's group. So this was kind of an add-on to my dissertation and something that Dr. Sapir wanted measured on some of the pilot scale bioreactors. And so it was one of those opportunities that if you are a PhD student, that you kind of have a core project, and then oftentimes you're asked to find a project that might be outside the direct scope. So this was definitely still in the scope of my overall dissertation, but was a really nice addition. And so these two papers that Emily Martin, also a graduate with a master's in using the pilot scale bioreactors, but these are two of my two most cited papers out of my dissertation. And so this work had an impact and helped us begin to answer some questions we have about hydraulic residence times, retention times, and wood chip bioreactors. So I'm not going to spend much time with this group talking about, what wood chip bioreactors are, but again, just that concept of reducing nitrogen loads to streams through this enhanced denitrification, hopefully to improve drinking water quality and reduce, hypoxic zones to the Gulf of Mexico. So we did have a number of questions, though, in regards to if this arrow or the amount of flow that's moving through a bioreactor is really big or really small, and the retention time of what is going on in terms of nutrient removal if you have a lot of flow or not a lot of flow. And so those two big questions in general were, how does nitrate removal differ among different hydraulic retention times? And then also, does HRT impact greenhouse gas emissions, specifically looking at nitrous oxide and methane? So I'm sure many of you are familiar with the pilot scale bioreactors. They are not currently in this form, as they were excavated, or some of them were excavated, and different media was put in after I left. I was lucky enough to continue working on some of that data collection as well. But we were looking at 3 different hydraulic retention times of 16 hours, 2 hours, and 8 hours. And we had these blocked off in 3 reps for a total of 9 reactors here. So for a couple years, we went out and we measured greenhouse gas emissions that were coming from the surface of these bioreactors, but also that were dissolved in the water moving through the reactors themselves. So we would sample the inlet, this well labeled A here, B, and then also the outlet, while also measuring what was coming off of the surface of the reactors. It's important to note that these reactors all had a soil cap on them, and we'll talk why that's important in the next few slides here. But so some of our first findings, and this was the focus of Emily Martin's work, was we found that we had a greater mass removal in our 2-hour HRT reactors compared to our 8 and our 16. So we saw and this is really highlights this importance of, are we trying to reduce concentration, or should we be focused on mass removal or load of nitrate left? So if you only looked at concentration, you could come to the conclusion that our 2-hour HRT reactors weren't working very well because they weren't dropping down this concentration, but they were actually removing the most amount of nitrate from the system. And so as it moves slower, we got a further reduction in this concentration, but less total removal. So that was the nitrate side of things. If we look at what happened in terms of greenhouse gas emissions, our x-axis here is essentially this cross section of the length of our bioreactor, and then these are, if we look at our flow past this point, is our load of our methane on the bottom here and nitrous oxide on the top. And so we can see that nitrous oxide tended in our 2-hour HRT to continue to increase all the way to our 4-meter, our second well, this well B, whereas our slower moving reactors, or the water moving slower, peaked at that first well, and then there was actually nitrous oxide consumption. We'll talk about why it was consumption here in a second when we look at some of our what we found coming off the surface. However, methane was close to the opposite, where our slower reactor, our 16-hour HRT reactor, was producing the most methane, and then our 2-hour kind of the least. And we bounce around here a bit, but this is expected, right? Because if we look at that, these anaerobic properties of the gases we would expect to be produced at different anaerobic states, we're looking at more anaerobic states for our methane to be produced compared to our nitrous oxide. So the other big finding from this work was that the bulk of the emissions of nitrous oxide and methane were not from the surface of the reactors, but what was dissolved moving through the outlet. So these are nitrous oxide production numbers, and we can see that we had significantly more nitrous oxide from our 2-hour compared to our 8 and 16-hour. They were not significantly different from one another. And then if we look at methane, we had our 8-hour and our 16-hour were significantly more than our 2-hour HRT reactors. So one way to bulk these greenhouse gas emissions together of both methane and nitrous oxide is to put them in a CO2 equivalent. So using global warming potentials of comparing nitrous oxide to methane to put them into grams of CO2 equivalent. And what we found here is that, again, it's just a different way to look at our results of the 2-hour HRT producing more total greenhouse gas emissions compared to our 8 and our 16. And so nitrous oxide has a global warming potential that's about 300 times that of CO2, whereas methane is only about 32. So that's why this nitrous oxide bar is so large for our 2-hour HRT. So finally here we tried to plot this to make some sense on, okay, what would be kind of an ideal HRT that, because we're removing the most nitrogen, the most nitrate at our 2-hour HRT, but we're also producing the most nitrous oxide. So if our nitrate removal line is more of a linear relationship with HRT, which is closer to that, you know, how far, what's the flow that we need to do before we really start seeing a significant uptick in our nitrous oxide emissions? So so here on our y-axis, we have this grams of CO2 equivalent per kilogram of nitrate removed, or excuse me, per gram of nitrate removed, and then we have our flow per day here. So I think I'm getting close on time here. So our hydraulic retention time. So this has brought about a number of newer questions, and some of those have been examined a little further in other manuscripts that have cited our manuscripts, Emily and I's manuscripts, and but really trying to kind of apply this to the real world of, because we don't have constant flow in the real world into bioreactors, and so are there any design features that we could use, like baffling water, to either slow it down or speed it up, depending on our desired nutrient removal, and then also nitrous oxide output. So I just wanted to end on kind of how this work helped me get into my current position. And you know, the more I was thinking about this when I was putting the presentation together, it was you know, kind of amazingly vital, and it was mainly because of the people that I got to work with with the Nutrient Reduction Center. So a lot of the bulk of my mentors, that people I really looked up to and weren't always necessarily on my committee, I was able to find through this group. And it's a group of people that are quick to accept grad students as peers and take your ideas seriously. And so there's so much value in that, of just giving you kind of confidence to go into an interview. But even programs like this, where you have to present your material in front of others, is very, very vital. So you have kind of this double end of really great mentors that are working with the center, as well as research ideas that are often cutting edge, or going to be applied not just in Iowa, but across the world, really. And so those were really just some valuable assets that I don't think I could have got anywhere else. And you know, like I said, if INRC wouldn't have funded some of this work, a couple of these papers wouldn't have been in my dissertation, and I wouldn't have the citations, et cetera, that are helping me move forward today. So, okay, hopefully I didn't go over too much. I can.
Perfect time.
Okay.
Are there any questions for Morgan? God, he won. Anybody online have a question? Are you okay? Are you still doing any bioreactor work down there, or is there interest in bioreactors or some of these edge of field practices in Missouri?
Yeah, that's kind of a tough one. So Kelly Nelson, who is housed up at in Novelty, Missouri, so kind of by Kirksville, if you're familiar with that. So it's essentially southern Iowa is what northern Missouri is. So the roads are a little worse than southern Iowa, is how I would put it. But he does have a kind of a dual system of a saturated buffer flowing into a bioreactor. And so we have, me and Dr. Kabir Singh are writing up some of that data. When I first got here, I got in contact with you know, some of the EPA and some of the point source groups down here. and there's not quite the funding for that, but I think it's only a matter of time, just from what I see in terms of tile that's going in, especially in northern Missouri. And so I've brought it up a few more times in terms of research questions, and I haven't gotten a ton of traction, to be honest, but I'm still kind of pursuing it with the Missouri Soybean Association down here, because I've kind of talked to them a lot about what is going, they already know what's kind of going on in Iowa. So it would be nice to get a few more of these in northern Missouri, but it's definitely slow, very slow moving. So.
All right. Well, in the interest of time, thank you so much, Morgan. And, I think speak for a lot here that many are proud of your accomplishments and excited what you're contributing to the scientific community as well. I don't want to ask how many years ago you first visited Iowa State, because that's going to make me feel really old. So yeah. I'm not going to ask that. Yeah. It probably was about 10 years ago.
Yeah.
Yeah, okay.
Yes, it was 10. Yes, a decade, which seems a little crazy to me. So I have, yeah. Then I didn't have gray hairs, and now I have a lot. I know they're just hidden in here. So
yeah. Well, yeah. Okay. Yeah. I think when we met the first time, I had hair. So yeah. So okay. We will turn it over to our next presenter, which is Prathyusha Cheguri. I apologize for that, but we're going to hear about a little more about one of the projects that Marshall mentioned, which is the perennial ground cover, PGC, and from what's happening in the field. Okay, I'm going to hide that. Okay. How do I hide the more? Yep. There we are. Hide video panel. There we go. Okay. Turn it over to you. And here, okay, here's a laser pointer if you want to. Okay. That way. Yep. And that's the speaker. So you can really speak up so they can hear you online.
But they can't see me.
They can see you. Yeah, that's that. Yep. They can see you. Online can see you.
Okay. So hello everyone. I am Prathyusha Cheguri. I'm a second year PhD student in Interdepartmental Genetics and Genomics program. I'm working with Dr. Xu Zhengfei in Department of Horticulture. Now I'm going to present about cool season grasses as perennial ground cover for sustainable corn production. The overview would be in this way. First, I'll go to the context and background, experimental design and data collection, results and discussion. Though I didn't do statistical analysis yet because I didn't get the yield data yet, I'll show some general trends with the traits I've collected data, future research implications, and then questions. Okay, so years ago, the native land cover of Iowa was mainly prairie. The rich soil, which is now enabling Iowa to produce so much corn and so much soybean, has only become rich because the soil was grown undisturbed for several thousands of years with continuous soil cover. And the precipitation is also largely absorbed by the soil by then because of rich organic matter. So the organic matter was built up by grasses and leaves through ages. And even though when runoff did occur because of this native vegetation, there was no runoff of water and nutrients. So because of these virgin conditions, by then the maximum protection was provided for the soil against erosion. But the things are different now because of the growing population, and also the increase in food demand is also made farmers practice monoculture agriculture, so which is a simple solution and high yielding one where cultivate one and eliminate the rest. So, but these monoculture agriculture has some multiple unintended environmental impacts, like it upsets the balance in the soil, robbing most of the soil nutrients and also decreasing the bacteria or microorganisms which are necessary to build up the organic matter. So one of the reasons for this soil erosion is the soil is left bare, mostly after the harvest and the spring, and the native land cover is destroyed and resulted in soil erosion, and intensive cropping resulted in decreasing organic matter and reduction in water absorption capacity, and also increase in water and nutrients runoff, mainly nitrogen leaching. This long periods of bare soil must be eliminated if we want the soil to be healthy. So, but how? The primary solution would be cover crops. But again, cover crops are not that, I mean, they're ideal, but they have some drawbacks, like they complicate the planting and harvesting dates and create risk of failure of both the crops. And also, whether the cover crop is successful or not, it requires high time and money every year. So I'm working on a very innovative approach called perennial ground cover approach, where grasses are intercropped with corn in between the corn rows. So this may have the advantage over the annual cover crops, like there'll be minimum additional inputs every year, continuous soil protection, year-round weed suppression and ground cover, and this may also reduce soil erosion, water runoffs, and nutrient runoffs, and build organic matter. But when besides green grows, there'll be obviously competition, and careful management of competition between these two main crop and the grass species is required. Mainly the competition would be for resources, that is soil moisture and nutrients, or interference like allelopathy, where grass may produce some harmful chemicals which may harm to the main crop, and also shade avoidance response, where the corn grows taller in response to the light quality. It won't get sufficient light quality and there'll be more vegetative growth. So we need to find a compatible PGC for this PGC-based cropping system. So the idiotype of compatible PGC would be cool season growth, shallow growth, rhizomatous and stoloniferous growth habit, shade tolerance, winter hardiness, and summer dormancy. I will be particularly talking in detail about the cool season growth and the summer dormancy. So why cool season grasses? Cool season grasses, when we compare to the corn, the C3 and like cool season grasses like cool and moist weather. Corn likes like summers and hot weather. Cool season grasses follow C3 photosynthesis pathway and corn follows C4, and the rooting patterns are also different. Cool season grasses have shallow root system compared to the main corn. So this is like choosing cool season grasses is one of the efficient way to minimize the competition. I also want, is this visible? Okay. I also want to tell about the importance and types of dormancy here. Dormancy is defined broadly as a temporary suspension of visible growth of any plant structure containing a meristem, which is an adaptive response to harsh environments. So dormancy is mainly categorized in three types: paradormancy, ecodormancy, and endodormancy. Paradormancy, it is mainly caused by some signals which are produced by the own plant parts from the distal plant parts. For example, apical dominance. Ecodormancy is a type of dormancy where the plant goes dormant and it comes back when the favorable conditions are provided. It comes back immediately. But endodormancy is something driven by internal biochemical signals. Even when the favorable conditions are on, the plant won't be active immediately. It strictly waits for sufficient time in dormant state. So the objective of this trial is to identify the best species or varieties, maintaining adequate ground cover with minimal reduction in the corn yield. These are the entries I am using. Okay. So 10 Kentucky bluegrass varieties and 1 Poa bulbosa and control. So till last year, 12 entries are used. This year, 3 additional entries are added to the same trial. That is ABC, Poa trivialis, 2 cultivars of Poa trivialis and 1 Poa secundum. This is a picture of the INRC trial. at Sorensen Farm, where corn is intercropped with grasses, and there's strip tillage, and then corn is planted in the rows here. I also want to go through about the growth pattern and morphology quick of these 4 different species. Poa pratensis, which is a Kentucky bluegrass, it reproduces as seeds and rhizomes. It is a long-lived perennial, and the dormancy here is ecodormancy. And the causal major driving force of this ecodormancy is the soil moisture. And next is Poa bulbosa. It reproduces as basal bulbs, bulbil, and rarely true seed. It is a short-lived perennial. The dormancy here is endodormancy, and the major driving factor here is photoperiod. And Poa secundum, it produces a seed. The dormancy is ecodormancy. Here the cause is higher temperatures, hot temperatures. Poa trivialis, it produces as stolons in seed. It doesn't go completely dormant, but it is a species where it is less tolerant to heat, and it is less active in summers. These are the data traits I collected: emergence count and final stand count, red floret ratios, plant height, staging, corn staging, chlorophyll spad readings, and grain yield. I'll be collecting later. These are the results of the previous student, Patrick, who has worked on the same trial when there are only 12 entries instead of the 3 extra entries this year. These are the results of 2020, 21, and 22. The yellow bar is the control, green is the Poa bulbosa, and the rest are the varieties of Kentucky bluegrass. So in almost all years, the yield of Poa bulbosa is compatible to control. And in the 2021 year, it have completed the control. And then 2022, all the yields are almost similar, because that was a season where there are a lot of floods and high water content and less nitrogen in the soil. So that's why the yields are low compared to other years. So this is basically what he did and summarized, is that he did the soil profiling, and he summarized that gravimetric water content is the major driving force for all the Kentucky bluegrass varieties to show lower yields compared to the control. This is plant height data of this year. The red bar is the control. Here is my pointer visible. Okay, this dark blue bar is Poa bulbosa, which is ranking next. And these are the 2 new entries which are added here. They're also doing good. And it may be because of their first year, or maybe they're really doing good. So these are the Poa trivialis and Poa secunda. This is the picture to show the height difference. The control, where the rows are not planted with any grass, it is just soil. You can see their height. They're my lab mates who helped me in collecting plant heights. This is the plot with Poa bulbosa. As we can see, it is little lesser height compared to the control. And this is one of the plot with one of the Kentucky bluegrass varieties. And this height is even shorter compared to the control. These are chlorophyll spad readings. This is an interesting one, where the control is the red bar, and this one, Poa secunda cultivars, have little outcompeted the control in the spad readings. And these last 3 are the 3 new entries which are added this year, and the Poa bulbosa. These are almost on par with the control. These graph is about the greenness ratings. So I have followed greenness ratings of 1 to 10 scale, where 1 is given for the complete brown or dormant one, and 10 is for the bright green one. So this orange curve here is for Poa bulbosa, which has gone dormant, completely dormant around 12 June. And the next is Poa secunda, which was also started getting dormant, like in the race. It stands second in the race after Poa bulbosa, and it has gone completely dormant around 10th July. And now these are all coming up. They're turning green now. And these 2 are the newly added Poa trivialis species. And all the curves up here are for the Kentucky bluegrass. So the driving force for the Kentucky bluegrass is the soil moisture. So there was very hot summers in this period in August. That's why there's less moisture, there are no rainfalls. And then they are dormant now. And most of them are dormant now. This is the picture of Poa bulbosa plot, which has started getting dormant around May 23rd. So the corn are just in seedling stage, and the Poa bulbosa, it is just turned from bright green, it has started getting dormant. And this is the same plot where Poa bulbosa got almost completely dormant around 19th June, in and around 19th June. So there are some green things here, are some broadleaf weeds. This graph is about corn staging. I mentioned this here because I found some interesting thing in the staging. This is the scale of, so corn staging starts from BE, emergence, and then V1, V2, V3 till the fully collared leaves. Every new collared leaf, so that would be assigned as next VN. And here around, before they get into R1 stage, the control, the Poa bulbosa and the Poa secunda plots have the stage higher compared to the other Kentucky bluegrass. If there are in like 8 or 9, if the Kentucky bluegrass is in 8 or 9 stage, the control Poa bulbosa and Poa secunda are at around 11, 12, and 13. So this, when we correlate to the plant height, it says that the higher stage ones is obviously little taller than compared to the Kentucky bluegrass varieties. The summary, like I have listed the rankings of the traits. Plant height, the first is the control, and next in the rank is Poa bulbosa, then Poa secunda, and Poa trivialis, the 4th place. For the chlorophyll SPAD ratings, the first rank is Poa secunda, which is outcompeted the control, and then Poa trivialis and Poa bulbosa. Next, the greenness ratings. Poa bulbosa is the first which to get dormant, followed by Poa secunda and Poa trivialis. These didn't go dormant completely, but they are always in the ratings of 3 and 4 when during the peak dormant period of Poa bulbosa, like 3 to 5, something like that. Then staging. The first one has around B14 stage, so it is in the first. The control is in the first rank. Then Poa bulbosa, Poa secunda, and Poa trivialis. So staging and plant height are almost correlated in the ranking. So conclusions and future research. So overall, this by using around 15 entries, mostly the Poa bulbosa, secunda, and trivialis showed like much variation, like on par with the control. So exploring more varieties of Poa bulbosa, Poa secunda, and Poa trivialis for increased compatibility into PTC system would be good. And I am working on other project where I work with Poa bulbosa at USDA accessions and do GWAS and like genome-wide association studies and RNA-seq to find the candidate genes for identifying, like find candidate genes for the summer dormancy trait in Poa bulbosa. Yeah. I would like to thank my professor, Kenmore, Raj Raman, Roger, and Patrick. Roger and Patrick were helping us throughout the field activities. Thank you.
All right. Questions?
Yeah. Is this primarily for like continuous corn cropping systems? And I was wondering if you were to rotate it with soybeans, would the same variety provide similar results?
Yeah, for now we are working on corn. So there are other trials where they're working on soybean as well. So for now, if we establish something in corn, then we can use the same thing for soybean and check.
Great.
How readily, okay, so Kentucky bluegrass, we can get seed, right? How readily available is with Poa bulbosa seed or the other Poa trivialis and that?
I am not completely sure, but for bulbosa, there's one company called Radix Hybrid bulbosa company. Only so far in USA, it's the only one producing that. I guess for bulbosa seed is also high. In future, we can make some hybrids and release some new varieties. Alright.
Where would pole bulbosa be grown right now? Where is it native to? Any
So it
idea?
is like Eurasia, mostly.
Matt was asking where it is produced, right? Not original.
No, I meant where, that's what I meant where it would be native.
Oh, native.
With native,
Okay,
so,
sorry.
yeah, I was just curious where it might be native.
On your greenness, that graph you had on your greenness rating, how are you measuring that? Sorry, I'm on my spec.
Okay. It is a visual estimation.
No, the previous one.
The greenness.
I was curious on this one too.
Is it about this graph?
No, the one, I think it was the one before.
SPAD readings.
No, with the line graph, sorry. Hang on up.
This one.
No, up. Forward to that. Yeah, go up. All right. All right.
That's it.
Oh, you're going the other way.
I mean, there are no other graphs.
Yeah. Okay, let's try it. Let's try, see. Anybody online, sorry if you're getting sick.
Right.
That one?
This one.
No, there was one before that.
Yeah, I
So
think
I
it was, I think it was this one now.
Greenness.
This is,
Yeah.
that's the greenness one here.
Oh, it's at the top. Yeah, I was looking to the heading. Yeah. Okay, sorry, my bad. How are you measuring that greenness?
Okay, so it's a visual estimation where I have given a scale of 1 to 10. 1 would be like completely brown and 10 is like bright green. So every week I used to go and take the ratings of each plot. For bulbosa, earlier around May or something, I kept a rating of, on 2nd June, I gave a rating of 3, where it has started getting dormancy around 3rd week of May. Then I gave a rating of 3, and then when it gets completely dormant, where there's no green at all observed on the soil, then it is 1. And for other Kentucky bluegrass varieties, they are still bright green at that time, and I've given 10 and 9, 8 ratings based on visual estimation.
So would it be good to the chlorophyll extract from that?
Oh, on ground cover?
Yeah.
Maybe.
So,
Yes.
And will you measure that through the fall to see when it comes out of dormancy, how?
Yes.
okay.
So the poor bulbosa already started
Is
becoming
it?
green.
Okay, okay. All right, let's
Are
thank.
there any
oh,
questions?
Oh, yeah, any questions online? I don't think there are yet, but if anybody wants to unmute and has questions. If not, let's thank speaker again. Appreciation, a coffee mug or a beverage mug of your choice. And we got 2 more that, depending on the, okay. John Kovar's already got one, so I'm gonna have John and Mitch. Did you get one too?
I got one.
Okay, all right. I'm taking ones.
I just used it today.
coming back.
No, okay. Here, I'm gonna let John Kovar draw. You're gonna draw the name. You get okay.
John's name is in there.
It is okay. I don't want one. Gee.
Oh,
Yeah, one more. One more. Yeah, we got one more here. We don't want to have to take it upstairs. Ryan. All right. Ryan's next door, and I haven't given him one. So, thanks everybody for attending, and come back in October because we're going to hear Ag Econ grad student and Lindsay Hartfeld that's at Wisconsin Discovery Farms now. So, thanks everybody for coming. Thanks to you online. Thanks, Matt. Oh, it's good. What's your name? Rob. John, have you met Ryan? Hey, thanks, Morgan. Sorry.
Thanks, Matt.
We'll
Have a good one.
send you a coffee mug.
Oh, yeah. Okay. Won't turn that down.
Yeah, okay. All right.
See ya.
Take care.